Today we are going to discuss about sewage treatment by microbes. Microbes are used in sewage treatment. Here nowadays in cities and towns, here the population is increasing and the sewage water, municipal water is also here it is released into the water bodies sometimes directly. So if it is directly released into the water bodies, so that is polluting the water bodies like rivers and streams. So that's why here before the disposal or before the release of this sewage water or municipal water into the rivers, here it is treated with microbes. So here the microbes are used in sewage treatment plants. Here STPs, sewage treatment plants. So the sewage treatment plants are generally they are constructed outskirts in the cities or towns. Here the sewage treatment, here it is completed in two steps. First one is here primary treatment. So here it is completed in two steps, primary treatment and secondary treatment. Here this is the sewage treatment plant. Here primary settling tank, aeration tank and secondary settling tank. First of all, the sewage is released into the primary settling tank. In the primary settling tank, here first of all the small pebbles and large pebbles and solid materials, here so much organic material is also present in the sewage. So all these are removed. So the organic matter and small pebbles and solid materials, these are removed or disposed in the sewage treatment plants. So the sewage is here it is collected in the primary settling tank. In the primary settling tank, here the large pebbles are the floating materials. The floating materials are here manually they are removed. So it is a sequential filtration. In the primary treatment, it is the first step again, sequential filtration. The sequential filtration in this the floating material, it is completely removed from the sewage water, and next here the grit is removed. The grit is removed by sedimentation process. Here by sequential filtration, the floating material, floating debris or floating material is removed. And by sedimentation method, the grit is nothing but soil and small pebbles. They are also removed from the sewage water. And some particles, some solid particles settle in bottom of the tank. The solid materials which are settled, they are also removed by sedimentation process. Here solid materials and grit. Grit is nothing but soil and small pebbles. So grit is soil and small pebbles. Grit is removed and solid materials are here they are settled, so they are also removed. And next the floating substance is called as effluent, primary effluent. It is a primary effluent. Here the settled particles are solid materials are they are removed. Here it is called as primary sludge. The primary sludge is it is released for disposal. It is disposed. And next the floating materials are the effluent, primary effluent. Here primary effluent is present. So this primary effluent is containing organic material and sometimes organic material and microbes are also present. a number of pathogenic microbes. So the organic material and pathogenic microbes, they are released or they are pumped into the aeration tank. So in the aeration tank, here sequentially or here aeration is, it is air is pumped. By this process, here the aeration tank, oxygen is pumped continuously. So the primary effluent is containing organic material and pathogenic microbes. So that is collected in the aeration tank, and here air is pumped continuously. Why air is pumped? Oxygen is pumped. Here in the presence of oxygen, the organic material is oxidized by here some microbes. Here heterotrophic bacteria are present. Heterotrophic bacteria are present generally in the here sewage or in the here primary effluent. So the organic material is oxidized in the presence of here oxygen by heterotrophic bacteria. So that's why a sufficient oxygen is here supplied to the bacteria in this tank, aeration tank. So continuously oxygen is pumped. So here in this primary effluent, here primary effluent is collected in the aeration tank and air is also pumped. In the presence of oxygen, the organic material and organic material is oxidized. So if the organic material is oxidized, here that is becoming as a less polluted. If it is having more organic material, that is more polluted. If it is having less organic material, it is less polluted. So that's why here the organic material content is reduced in this aeration tank by using some microbes. Here the microbes are heterotrophic bacteria. Here generally they are present in the sewage. Here the BOD is reduced. Here BOD. Here the what is the BOD? BOD is biological oxygen demand. Primary effluent is collected into the aeration tank. Some heterotrophic bacteria are present and air is also pumped or oxygen is pumped. In the presence of oxygen, the organic material is oxidized. So it is completely secondary treatment. In the secondary treatment, the primary effluent is collected into the aeration tank. So here in the aeration tank, mainly the BOD is reduced, biologically oxygen demand. Here the BOD is nothing but the required amount of oxygen to oxidize the organic material present in the 1 liter of sewage or water by bacteria. Here the bacteria consume some oxygen to oxidize the organic material. So the required amount of oxygen is called as BOD, biological oxygen demand. If more organic material is present in the sewage, here more oxygen is required for the decomposition of organic material or for the oxidation of organic material. If it is having less organic material, it requires less amount of oxygen. So the sewage water, if it is having more organic material, that is more polluted. If it is having less organic material, that is less polluted. So mainly in the BOD is reduced in the aeration tank. So BOD is reduced. And next here the effluent is here in the aeration tank. Mainly the bacterial growth is taking place. Mainly bacterial growth. The bacterial growth is after bacterial growth, here flags are formed. Flags are nothing but bacteria and fungi. Here number of bacteria. Here in the presence of oxygen, the aerobic bacteria are growing and number of bacteria are growing in this aeration tank. So if more bacteria are present, here easily or rapidly the organic material is oxidized. So in the presence of oxygen, here the aerobic bacteria are growing in this aeration tank. And next fungal filaments are also present in the sewage. So the fungal filaments and the bacteria both are forming the flags or mesh-like structures. For example, here it is all these are fungal filaments. All these are the fungal filaments. Here fungal filaments and bacteria, they are forming the mesh-like structure. So the mesh-like structures are called as flags. So the flags are formed in the aeration tank and they are settled. Next here the total sewage is, here it is supplied into the secondary settling tank. In the secondary settling tank, here the total flags, here flags are formed in the aeration tank and this effluent is supplied into the secondary settling tank. So the flags are settled. Here all the flags are settled. So the settled flags together called as secondary sludge. Here settled sludge or secondary sludge. And the effluent is nothing but here clarified sewage water. It is released into the water bodies or rivers or streams. Next in this tank the settled flags are, here some are disposed. Some amount of flags or secondary sludge is disposed. And next some amount of sludge is or secondary sludge is, here it is again pumped into the aeration tank. So only little amount of secondary sludge or activated sludge. The activated sludge is pumped into the aeration tank. So this secondary sludge or activated sludge which is pumped into the aeration tank, it is acting as inoculum. Inoculum means starting material. Here if there is no bacteria, here no bacterial growth, no oxidation of organic material. That's why here first some amount of some bacteria should be present in the aeration tank. That's why here if all the bacteria are moving into the secondary settling tank, here if no bacteria are present in the aeration tank, the organic material in the aeration tank, it is not oxidized. So BOD is not reduced. That's why here some bacteria or activated sludge is again pumped into the aeration tank and a large amount of secondary sludge is disposed. Okay. Next the effluent is, it is released into the, it is less polluted sewage water. So the less polluted sewage water or clarified effluent is released into the water streams. So it is microbes in sewage treatment. Next after the secondary treatment, the secondary sludge, here the secondary sludge is large amount of secondary sludge, it is again pumped into the another tank, that is anaerobic sludge digester. It is anaerobic sludge digester. In this anaerobic bacteria are involving. Here the clarified effluent is released into the water bodies. So it is less polluted sewage water and the activated sludge is containing number of bacteria and pathogenic bacteria. Next fungal members. So the secondary sludge, it is taken into the anaerobic sludge digester. Here anaerobic bacteria are involving. In the anaerobic bacteria, generally these are methanogens. The methanogenic bacteria, methanobacillus, here methanogenic bacteria, they are here oxidizing or they are digesting the bacteria and fungal members. Here the flags are there. Here flags. Flags are nothing but bacteria and fungal members. So the here secondary sludge is containing number of bacteria and fungal members. So the bacteria and fungal members are here digested by anaerobic bacteria. So the bacterial cell walls and fungal cell walls, here generally they are containing cellulosic materials. Here the cell wall components are completely digested. So when the cell wall materials are digested, here the methane, mainly methane and carbon dioxide, hydrogen and hydrogen sulfide, these gases are released. So different bacteria are involving, but here based on the bacteria involving in this anaerobic sludge digestion, here the gas is also depending. Generally the biogas is containing here methane and carbon dioxide, hydrogen sulfide and hydrogen. So these are the main component in the biogas is methane. So the biogas is generally that is collected here and it is supplied to the nearby houses. So during the digestion of bacteria and fungal members, here the gas is released. Here one roof is there. So it is one roof. It is inlet and it is roof. The roof is it is rising. It keeps on rising on the sludge when the gas is produced by the bacteria. So during the bacterial digestion, the gas is released. So the roof is also rising in the level, and the gas is collected and it is supplied to the nearby houses. So the biogas is generally used as it is inflammable, highly inflammable. That's why it is used for cooking and lighting process. Here the biogas components are produced by during the digestion of bacteria and fungal member, the biogas is released. So that's why here in villages generally the dung is containing number of methanogenic bacteria. So these methanogenic bacteria are digesting the bacteria and releasing the biogas. So the biogas is produced in the anaerobic sludge digester during the digestion of bacteria and fungal members. So the biogas is released. So that is collected and used for the cooking purpose and lighting purpose also.